So imagine printing out your own genetic code, just, putting it all on paper.
Well, it would be massive.
Yeah, it would form a stack of books, like 100 stories high, which is insane. And somewhere in that giant skyscraper of text, there could be a single 6-letter typo that determines if your brain will deteriorate before you even turn 60.
It's terrifying when you put it like that.
It really is. But today, in this deep dive, we aren't just looking at how scientists find that typo. Reading through the stack of sources you sent over covering the 2026 Breakthrough Prize ceremony, well, it's clear scientists have literally figured out how to use chemical whiteout to erase that typo while a person is still alive.
Exactly. And it is a profound shift, in how we actually interact with nature.
Yeah.
The materials we're exploring today, for you listening, they really capture a moment where humanity is crossing a threshold. We're moving away from just observing the laws of physics and biology.
Right, just watching things happen.
Yeah, and we are actively stepping in to rewrite them.
The setting for all of this is just wildly contradictory, though, which makes your notes so fun to read. Picture this scene, right? You have the 2026 Breakthrough Prize ceremony happening on a Saturday night in Los Angeles.
In the heart of Hollywood.
Yes, the room is just dripping with Hollywood royalty. You have Michelle Yeoh, Robert Downey Jr., Margot Robbie. James Corden is hosting the whole thing.
And he had some pretty good jokes, actually.
Oh, he called it the billionaire's ball. And he was giving all these tech founders advice to avoid copying Robert Downey Jr.'s mustache.
Which is solid advice, honestly.
Truly. But you have this ultimate Hollywood glamour colliding with the most rigorous, universe-altering science on Earth. It's such a weird mix.
The contrast is stark, but the structure of the prize itself is what really sets the tone. These aren't your standard institutional research grant.
Right, where the money goes to the university lab.
Exactly. The Breakthrough Prize awards $3 million directly and personally to each winning scientist. It completely bypasses the university bureaucracy. It goes straight into their personal bank accounts.
Wow, just direct deposit.
Pretty much. And it's a very deliberate choice to honor the theme of the night, which was standing on the shoulders of giants. It's really about recognizing the individual human minds that, drag our collective understanding forward.
James Corden actually had a great bit about the collision of these two worlds. He joked that ChatGPT has completely eroded his ability to have an original thought.
I think we could all relate to that a little bit these days.
For sure. But he pointed out the irony that Hollywood is terrified of artificial intelligence because, they hate anything artificial. But underneath the joke, there's a real tension there. Hollywood is obsessed with preserving this specific static image, while the scientists in that room are dedicating their lives to just embracing the absolute unknown.
And that willingness to dive into the unknown is most visceral when we look at the genetics revolution that they celebrated that night.
Yeah.
The researchers honored are taking diseases that have historically been considered irreversible life sentences.
Like nothing you can do about it.
Right. And they're essentially just turning off the underlying mechanism.
Yeah, the sources outline the work of Swee, Thein, and Stuart Orkin. They targeted sickle cell anemia and beta thalassemia. Millions of people suffer from these conditions, right?
Millions, yes.
Where their red blood cells warp into these rigid sickle shapes. It causes immense pain and organ damage because the cells literally just can't carry enough oxygen.
It's devastating.
But your notes highlight a fascinating loophole in human biology. Babies don't have this problem.
They don't.
They produce something called fetal hemoglobin, which works perfectly. The disease only manifests later in life. So what exactly changes?
Well, the symptoms only start because of a biological timer. Shortly after birth, there's a genetic switch that flips.
Like a literal on-off switch in the code.
Pretty much. It instructs the body to stop producing the healthy fetal hemoglobin and start producing the defective adult version. So Thein and Orkin decided that instead of treating the symptoms, they needed to find that switch.
Oh, that makes so much sense.
Right. Through their research, they identified a specific gene called BCL11A. And this gene's entire job is to act as a suppressor. It shuts down the fetal hemoglobin factory.
So the logic is incredibly elegant. If BCL11A is the guard keeping the healthy factory closed, you just need to, remove the guard?
Exactly. Orkin proved that if you genetically silence that specific gene, the body never produces the defective adult hemoglobin at all.
Wow.
It just keeps the healthy fetal hemoglobin flowing indefinitely. And that exact discovery is what led to the very first FDA-approved CRISPR therapy.
The implications of that are staggering. And we see that same transformative approach in the work of Bennett, Katherine High, and Albert McGuire, right?
Yes, their work is incredible. They developed the first FDA-approved gene replacement therapy for an inherited form of blindness called LCA.
How do you even fix blindness with genetics?
Their mechanism involves taking a healthy copy of the missing gene, placing it inside a harmless modified virus, specifically an adeno-associated virus, and then they inject it directly behind the patient's retina.
So they use a virus as like a delivery truck?
That's a great way to think about it. The virus acts like a delivery truck, dropping the healthy code directly into the retinal cells, which then start producing the protein needed for sight.
Reading the emotional weight of this in the sources is something else. Like, they started the trials on blind puppies. Actual blind puppies that completely recovered their sight.
It's a heartwarming detail.
It really is. And that gave them the data to treat humans, including a little girl named Hannah Reef. Her father described her condition by saying she essentially looked at the world through 2 straws.
Just zero peripheral vision.
Right. Couldn't see anything in the dark, couldn't even see the stars at night. And then, according to the notes, she received the gene therapy at age 7. And her mother tells this story about standing outside shortly after the treatment. Hannah just points up at a tiny white dot in the sky and says, "I can see it." She saw a star for the first time in her life.
That's just amazing.
The sources note she was actually in the audience at the ceremony like a confident young woman who now rides horses. It's the ultimate proof of concept. But the notes also make it clear that for many genetic diseases, we don't have the luxury of time. Like, the clock is just ticking relentlessly.
That urgency is really the driving force behind Rosa Rademacher's and Brian Trainor's research. They were investigating 2 devastating fatal brain disorders, ALS, which degrades motor neurons, and frontotemporal dementia, or FTD, which destroys the frontal lobes of the brain.
And historically, doctors treated those as totally separate things, right?
Completely separate. But Rademacher's and Trainor noticed they occasionally clustered in the same families, suggesting a shared, hidden root. So they mapped the genomes and found it. It's a mutation known as the C9 repeat expansion.
Looking at the explanation in the material, this mutation isn't just a missing piece of code. It's almost like a stutter.
A stutter is the perfect word for it.
It's this tiny 6-letter sequence of DNA that just repeats over and over hundreds or thousands of times, and it creates these toxic proteins that destroy the brain, which, bringing it back for you listening, goes back to that analogy of the human genome being a 100-story stack of printed books.
Finding that repeating 6-letter typo in billions of letters is an unbelievable feat of mapping.
It really is. But knowing the typo is there doesn't cure the patient. Like, how do you actually go into the cells of a living human being and fix it without just destroying the surrounding code?
The answer to that mechanical problem lies in an unprecedented medical event detailed in the sources, the case of baby KJ.
Oh, this story is wild.
It is. Baby KJ was born with CPS1 deficiency, which is a fatal metabolic disorder. Basically, the liver simply cannot process protein, leading to a toxic buildup of ammonia in the blood. The fatality rate is incredibly high, and the root cause is a single misspelled letter out of the 3 billion letters in his genome.
1 letter out of billions.
1 single letter. But a team, including Kieran Sunuru, Rebecca Ahrens-Nicholas, and David Liu, realized they could potentially fix it. But to answer your question about how we fix the typo without destroying the code, we have to look at the evolution of CRISPR.
Okay, lay it on me.
Traditional CRISPR acts like a pair of molecular scissors. It finds a specific sequence and cuts the double helix completely, relying on the cell to patch the break. But cutting the DNA can cause unpredictable damage or unwanted mutations.
So the scissors are too blunt for a single-letter typo?
They are. It's too risky. Instead, the team used a newer technology called base editing. If traditional CRISPR is a pair of scissors, a base editor is a pencil eraser.
Ah, I like that.
It doesn't sever the DNA strand. Instead, it uses a specialized enzyme to grab that single misspelled letter, say an A, and chemically rearrange its atoms so it physically transforms into a G. They literally rewrite the text without ever tearing the page.
The timeline of this is what absolutely blows my mind. Normal drug development, from concept to trial, takes what, 5 to 10 years?
Easily.
But baby KJ was dying. So this team collaborated with Peter Marks at the FDA to just shatter every existing bureaucratic speed limit. They engineered a custom, personalized base editing drug for this one specific infant in 6 weeks.
6 weeks from concept to reality. It's unheard of.
And the parents were faced with the impossible choice to inject an entirely untested, first-of-its-kind medicine into their newborn.
But it worked. Within 10 days of receiving the personalized drug, his body began processing protein normally. It was the first time in history a patient received a personalized gene-editing drug.
Wow.
The medical community described it as a moon landing. We have definitively proven that genetic destiny is no longer an immutable law. It is a negotiable draft.
That is profound. And Yentley Soto Albrecht's speech at the ceremony perfectly captured what that means for a human life. She's a newly minted MD PhD, and she lost her father to ALS in just 14 months.
That's so fast.
And she knows she inherited the exact same C9 repeat expansion mutation we discussed earlier. Statistically, she has a 95% chance of developing ALS or FTD by age 65, but rather than resigning herself to that fate, she is volunteering her body, her blood, her spinal fluid, and her cells to accelerate the cure.
Yeah, she said Raidmakers and Trainer mapped the battlefield, and now she is going to wage the war.
Just incredible courage.
Our resilience is extraordinary. But we keep talking about editing DNA as if it's a static string of code sitting neatly on a desk.
Right, like an actual book.
Exactly, but it isn't. This code is floating inside a living, chaotic, constantly churning cell. How does a cell actually organize all this editing, transcribing, and protein building without just turning into a microscopic soup?
This is where the source material completely upends basic high school biology. The Breakthrough Prize in Life Sciences went to Cliff Brangwyn and Tony Hyman. For decades, textbooks told us that a cell functions like a walled factory.
Right, with all the neat little compartments.
Yeah. You have the nucleus for administration, the mitochondria for power, all neatly separated by physical membranes. But Brangwyn and Hyman looked closer and realized that massive amounts of cellular work happen outside those walled structures.
They discovered a phenomenon called the phase separation of proteins and RNA. Under specific conditions, fluids inside the chaotic environment of the cells spontaneously condense into fleeting, membraneless liquid droplets.
So the inside of our cells operates more like a microscopic lava lamp.
That's exactly it.
You have these distinct blobs of fluid merging together to perform a task and then separating and dissolving back into the background when they're done.
The lava lamp visual captures the mechanics perfectly. These liquid droplets form spontaneously due to the chemical interactions between specific proteins and RNA. They pull themselves together into concentrated hubs, allowing cellular agents to meet, initiate chemical reactions, express genes, and then disperse.
It's so fluid.
It completely challenges the fundamental structural rules of biology. Organization in a living system doesn't always require physical walls. Sometimes it just requires the right chemical density.
Now, if biologists are finding hidden spontaneous order inside the messy fluid of our cells, the sources highlight how mathematicians are doing the exact same thing with pure numbers.
Yes, the Breakthrough Prize in Mathematics went to Frank Merle. He studies nonlinear evolution equations. These are the mathematical models we use to predict wildly turbulent systems.
Like what kind of systems?
We use these equations to map things like rogue waves in the ocean that appear out of nowhere to swallow ships, or a single spark that rapidly swells into an uncontrollable inferno.
The most fascinating example from the notes is a laser beam shining through glass. It sounds simple, but the mechanics are chaotic. When you shoot a high-intensity laser into a medium like glass, the intense light actually alters the refractive index of the glass itself.
Right, it changes the physical property of the glass.
Yeah, the glass essentially turns into a self-focusing lens. That lens focuses the beam tighter, which increases the intensity even more, which makes the glass an even stronger lens. It becomes this runaway feedback loop until the beam's brightness spikes toward infinity and the glass literally shatters.
And in mathematical terms, that sudden, violent spike to infinity is called a blow-up. For a long time, mathematicians struggled to map exactly what happens inside that feedback loop. The equations become overwhelmingly chaotic as they approach infinity.
Because the numbers just get too wild.
Exactly. But Frank Merle found a way to map the chaos. He discovered a specific threshold, a fraction of a millisecond before the blow-up happens. And right at that critical boundary, hidden deep within the turbulent mathematics, he found a simple, perfectly stable wave. He found an elegant, predictable structure anchoring the absolute center of a chaotic event.
But while Frank Merle is hunting for stability hidden inside a chaotic equation, the physicists at this ceremony were doing the exact inverse. They took the most perfectly stable, predictable equation in human history and desperately tried to break it.
They're targeting the standard model of particle physics. The standard model is essentially the master blueprint of the universe. It perfectly predicts how the fundamental forces in particles interact from the subatomic scale all the way to the cosmic scale.
It's like the holy grail of physics.
It is. Its mathematical predictions align with physical reality with flawless precision.
And yet, you have 283 scientists from CERN, Brookhaven National Laboratory, and Fermilab who spent 60 years trying to find a flaw in it.
Talk about dedication.
Seriously. They won the Breakthrough Prize in Fundamental Physics for the muon G-DEFTA experiment. Looking at the setup, it feels like science fiction. They use a particle called a muon, which is essentially a heavier cousin of an electron. It only exists for a millionth of a second before decaying.
Blink and you miss it.
Right. They shoot these muons into a 50-foot wide ring of superconducting magnets and track them as they zip around at nearly the speed of light.
And as the muon travels around the ring, it also spins on its own axis, acting like a tiny, microscopic bar magnet. When it passes through the magnetic field of the ring, the axis of its spin wobbles, much like a spinning top wobbles as it slows down.
Okay, I can picture that.
The standard model mathematically predicts the exact speed and angle of that wobble.
And the reason the standard model predicts it so precisely is because the vacuum of space isn't actually empty, right?
Exactly.
The notes explain that as the muon spins, virtual particles are constantly popping in and out of existence, bumping into the muon and altering its wobble. The physicists measured that wobble to a mind-boggling degree of precision, and they found a discrepancy.
Like a tiny one.
Yeah, the muon wobbled just a microscopic fraction of a percent differently than the standard model predicted.
A discrepancy that small might seem like a rounding error. But in physics, a tiny crack in a perfect theory is the ultimate prize. How? Think of Isaac Newton's theory of gravity. For centuries, it flawlessly predicted the movement of the planets. But there was a minute discrepancy in how Mercury orbited the Sun. Its orbit shifted by just a fraction of a degree over thousands of years.
Just a tiny wobble of its own.
Right. It seemed like nitpicking. But Albert Einstein realized that tiny anomaly meant Newton's entire framework was incomplete, which led him to invent general relativity. The muon discrepancy means the exact same thing. If the muon is wobbling at the wrong speed, it means it's bumping into undiscovered particles or forces in the quantum vacuum.
So the universe has layers we just haven't mapped yet.
Exactly.
The sheer thrill of knowing the map is incomplete is what drives all of this. And the ceremony highlighted another massive blank spot on the map: dark matter. Jensen Huang, the CEO of NVIDIA, announced the new Vera Rubin New Frontiers Prize.
Named after the legendary astronomer.
Right. Vera Rubin was the one who proved the existence of dark matter by showing that galaxies were spinning so fast they should fly apart, meaning there had to be some invisible mass holding them together.
And NVIDIA actually named their new AI supercomputer after her, demonstrating the growing link between computational power and theoretical physics. The inaugural winner of that prize, Carolina Figueiredo, was honored for discovering unexpected geometric connections between entirely different quantum field theories.
Basically simplifying how we calculate the interactions of particles.
Yes.
Speaking of how particles interact, the final massive physics award went to David Gross for decoding the strong force. The source material describes the strong force as a Romeo and Juliet style bond holding the core of the atom together, but what actually makes it act like that? Most forces, like gravity or electromagnetism, get weaker as you pull things further apart.
Well, the strong force operates on an entirely different mechanism called quantum chromodynamics. The particles inside a proton, called quarks, are bound together by particles literally called gluons.
Because they act like glue.
Exactly. Gluons carry the strong force. But unlike photons, which carry light and basically ignore each other, gluons actually interact and stick to each other. So when you try to pull two quarks apart, the gluons stretch between them like a thick rubber band. The further you pull, the tighter the rubber band gets, storing massive amounts of energy.
Until eventually the rubber band snaps.
Right, and when it snaps, the stored energy is so immense that it spontaneously creates entirely new quarks out of the vacuum, rather than letting the original two exist alone.
That is wild.
It is. Because David Gross decoded the mathematics of that rubber band effect, scientists were able to reverse engineer it. They realized that if you squeeze quarks close enough together, the rubber band goes slack, and the particles behave freely. This allowed physicists to reconstruct the quark-gluon plasma, which is the unimaginably hot, frictionless liquid state of the entire universe, just a millionth of a second after the Big Bang.
You have scientists mapping the literal dawn of time. And then at the same ceremony, you have Mark Rober, the YouTube engineer, stepping onto the stage in a dress shirt and no socks.
What a visual.
He stands next to a visibly terrified James Corden wearing safety goggles. Robert takes a massive container of liquid nitrogen, which sits at negative 320 degrees Fahrenheit, and drops it into a bucket of boiling water.
Oh, boy.
Yeah, the rapid expansion creates a 30-foot indoor cloud that completely engulfs the ballroom. It's a hilarious visual, but it makes you ask, amidst all this serious, universe-altering science, why do a giant stunt cloud?
Well, it serves a critical purpose. Spectacle sparks wonder, and wonder is the prerequisite for discovery. David Gross spoke eloquently about this in his acceptance speech. Curiosity isn't just a byproduct of science, it's the fundamental engine.
It's what gets them out of bed.
Precisely. And you see that engine operating perfectly in the story of Matea Canizares. She is a 17-year-old student from a rural town near Quito, Ecuador. She won a $250,000 scholarship in the Breakthrough Junior Challenge.
And what was her project?
A brilliantly animated video explaining the exact same quark-gluon plasma that David Gross won his prize for.
A high school student explaining the dawn of time. It proves that the human desire to decode the universe has no geographic boundaries. But your notes also highlight the brutal reality of this work. It requires a massive tolerance for failure.
Oh, absolutely.
Robert Downey Jr. underscored this when he took the stage to read an old high school report card belonging to the late great scientist Sir John Corden. RDJ read it in this ridiculous Chico Marx accent. Gurdon's teacher had written that his work was disastrous, that he refused to listen, and that his dream of becoming a scientist was quite ridiculous and a sheer waste of time.
And Gurdon went on to win the Nobel Prize for discovering that mature cells can be reprogrammed into stem cells. It is a powerful reminder that the scientific process is built on a mountain of failed experiments and dismissed ideas.
You have to push through the noise.
You do. And that theme of enduring failure was central to the tribute presented by Ron Howard, Brian Grazer, and Tom Hanks for Jim Lovell and the crew of Apollo 13.
The Apollo 13 story is the ultimate example of what scientists do every day in the lab. They were 200,000 miles from home in the vacuum of space. Their oxygen tank had exploded, their systems were failing, and they were freezing. Panic would have been completely justified.
But they didn't panic.
No. They showed absolute grace under unbearable pressure. They just kept working the problem. Whether it's a broken spacecraft, a wobbling muon, or a typo in a piece of DNA, the job is to work the problem.
Which brings us back to the sheer scale of what this ceremony celebrated. The scope of human achievement documented in these sources is breathtaking.
We have covered so much ground today. We started at the absolute micro level, exploring how base editing acts as a molecular pencil erasure to rewrite single typos in the human genome, saving infants from fatal metabolic diseases. We zoomed out to see how the chaotic fluid inside our cells spontaneously forms microscopic lava lamps to organize biological life.
We watched mathematicians find elegant, stable jewels right before a laser shatters a piece of glass.
And we looked at physicists hunting for undiscovered particles by tracking the wobble of a microscopic magnet, while others reconstruct the plasma from the dawn of time.
And tying all of these breakthroughs together is a rapidly accelerating variable. Throughout the ceremony, computing power and artificial intelligence were repeatedly highlighted as the new primary instruments of discovery.
The new telescopes, basically.
Exactly. Jensen Huang described these AI systems as time machines that allow researchers to simulate the evolution of entire galaxies and decode biological proteins in a matter of days tasks that used to require entire lifetimes.
It's moving so fast.
It is. But as we synthesize this hyper-advanced computational power with our newfound ability to mechanically rewrite human biology and reverse engineer the Big Bang, we are faced with a profound paradigm shift. Well, what happens when the technological tools we originally built to simply understand nature become sophisticated enough to completely invent it? For you listening, something to really ponder. Are we culturally and philosophically prepared for a future where we aren't just standing on the shoulders of giants, but we are actively playing the role of the giant?
That is an incredible thought to leave on, because if reality is a 100-story printed book, we are no longer just reading the pages. We aren't just using an eraser to fix the typos either. We are picking up the pen, and we are starting to author the next chapter ourselves. Thank you for joining us as we explore these sources today on this deep dive. Keep working the problem, keep looking closely at the chaos, and keep your curiosity alive.